Hello there, and welcome to the Sleeping Physicist. If you are someone who finds it calming to follow the structure of the world down to its smallest foundations, then you are in the right place. Here we take real physics slowly. We allow ideas to unfold carefully, without rushing past the details that make them beautiful. Everything we explore is grounded in established science. Nothing speculative, nothing exaggerated, only what patient observation and theory have revealed about nature. If you find yourself enjoying these unhurried explorations, you are welcome to like and subscribe. For now, allow your attention to settle. Let the day loosen its grip. Let curiosity guide us inward. What is DNA made of according to quantum physics? You have likely learned that DNA carries genetic information. It determines traits. It allows organisms to grow, repair, and reproduce. But before DNA is information, before it is biology, it is matter. It occupies space inside cells, it has mass, it has structure. If we could magnify a strand of DNA until it filled the room around you, you would see a long molecular chain shaped like a gently twisted ladder. Two rails run alongside each other, connected by evenly spaced rungs, and the entire structure coils into a spiral. This shape is not symbolic, it is physical. Inside a living cell, DNA is not floating as an abstract blueprint. It is folded, coiled, and packed into a confined space, held together by forces that do not care about life or meaning. The stability of that spiral form arises from interactions between charged particles. The repeating pattern of its structure arises from chemical bonds. Every curve and connection follows physical rules. At this first level of understanding, nothing mysterious is required. DNA is a molecule with a specific geometry. Molecules are arrangements of atoms. Atoms follow the laws of physics. If we want to understand what DNA is made of according to quantum physics, we must move inward step by step, from visible biological structure toward the smaller layers that make chemistry possible. For now, what is firmly true is this: DNA is organized matter arranged into a stable repeating form inside living cells, and that form exists because deeper physical laws govern how its smallest components hold together. When we look more closely at that twisted ladder shape, its structure begins to resolve into repeating chemical units. DNA is not one continuous piece. It is built from smaller components linked together in sequence. Each repeating unit is called a nucleotide, and each nucleotide has three parts: a sugar molecule, a phosphate group, and a nitrogen-containing base. The sugar and phosphate groups form the two long rails of the ladder. They alternate in a steady pattern: sugar, then phosphate, then sugar again, creating a backbone that gives the molecule its shape and strength. The nitrogen bases extend inward from this backbone. These bases form the rungs of the ladder by pairing with complementary bases across the spiral. Their arrangement encodes biological information, but physically they are simply molecular structures made from specific arrangements of atoms. There are 4 types of bases in DNA. Each has a slightly different shape and distribution of electrical charge. These differences determine which base can pair with which. Adenine pairs with thymine. Cytosine pairs with guanine. This pairing is not guided by intention. It arises from how their atomic structures allow certain interactions and forbid others. The geometry of the molecule emerges from these constraints. At this level, everything we see belongs to chemistry. Atoms connect through chemical bonds to form stable molecules. Those molecules connect in repeating sequences to form long chains. The double helix shape is a consequence of how these pieces fit together most stably in 3-dimensional space. Stability is not aesthetic, it is energetic. The arrangement that requires the least energy is the one that persists. So DNA, in chemical terms, is a long polymer composed of repeating nucleotides, held together by bonds between atoms and stabilized by interactions between its paired bases. The structure is determined by how those atoms share and distribute electrons. And because chemistry itself depends entirely on atoms, the next layer of understanding lies beneath the molecule, in the structure of the atom itself, where the rules governing those bonds begin. The chemical structure of DNA rests entirely on atoms. Every sugar unit, every phosphate group, every nitrogen base is composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus atoms arranged in precise configurations. Chemistry does not float above physics. It is the large-scale expression of how atoms combine. An atom appears, at first glance, to be a tiny solid particle. For centuries it was imagined that way, as an indivisible bead of matter. Modern physics reveals something quieter and more structured. An atom has a small, dense nucleus at its center. Around that nucleus extends a region occupied by electrons. The nucleus contains protons and neutrons. Protons carry positive electric charge. Neutrons carry no charge. Electrons carry negative charge and are much lighter than the particles in the nucleus. The size difference is dramatic. If the nucleus were scaled to the size of a marble placed at the center of a cathedral, the electrons would occupy a vast surrounding region extending almost to the walls. Most of what we call solid matter is structured emptiness governed by electric forces. The apparent solidity of DNA arises not from packed substance, but from interactions between charged particles. In DNA's atoms, the positively charged nuclei attract negatively charged electrons. At the same time, electrons repel one another. Protons inside the nucleus repel each other as well, yet they remain bound together by a stronger interaction that operates at extremely short distances. Even at this atomic level, structure emerges from a balance of forces rather than from rigid material. The atoms that compose DNA differ from one another only in the number of protons within their nuclei and the arrangement of their electrons. Carbon has 6 protons, oxygen has 8, nitrogen has 7. These differences determine how many electrons each atom can share or attract when forming chemical bonds. The behavior of DNA at the chemical scale therefore depends entirely on how electrons are arranged around atomic nuclei. At this stage, we see that DNA is made of molecules, molecules are made of atoms, and atoms are organized systems of charged particles governed by fundamental forces. The stability of DNA's chemical bonds depends on how those charged particles behave. To understand why atoms arrange themselves the way they do, we must look more closely at the structure of the atom and the forces that hold its components in place. Within each atom that composes DNA, there is a concentrated center that holds almost all of its mass. This center is the nucleus. It contains protons and neutrons packed into an extraordinarily small volume. Protons carry positive electric charge. Neutrons carry no charge, yet they contribute mass and stability. The protons inside the nucleus repel one another electrically, but they remain bound because another interaction operates there, one that is far stronger at extremely short distances. The strong nuclear force acts only within that confined space, and it is sufficient to prevent the nucleus from flying apart. Surrounding this compact center is the region associated with electrons. These electrons are not tiny beads circling along clean paths. When physicists examined atoms more closely, the classical picture dissolved. Electrons do not travel in fixed orbits. Instead, their presence is distributed. The best description available is probabilistic. An electron is found in certain regions with greater likelihood, in others with less, and these patterns are determined by quantum rules. The positive charge of the nucleus pulls on the negative charge of the electrons. This attraction gives the atom cohesion, yet the electrons cannot simply collapse into the nucleus. Quantum mechanics restricts the energies they are allowed to have. Only certain discrete energy states are permitted, and each allowed state corresponds to a specific spatial pattern. These quantized arrangements give the atom a stable size and prevent matter from compressing indefinitely. The atoms in DNA differ from one another because they contain different numbers of protons and therefore different numbers of electrons. Carbon, oxygen, nitrogen, hydrogen, and phosphorus each possess characteristic outer electron arrangements. Those outer electrons determine how many connections an atom can form and what shapes those connections prefer. The familiar patterns of chemistry emerge from these quantum constraints. Inside every nucleotide, then, there are nuclei bound by the strong force and electrons arranged in quantized distributions around them. The mass of DNA resides mostly in those nuclei. The shape and bonding behavior arise from the structured behavior of electrons. What appears to us as solid biological matter is, more precisely, a stable arrangement of charged particles held in place by fundamental forces that operate according to strict quantum rules. The stability of every atom inside DNA depends on electric charge. Protons carry positive charge. Electrons carry negative charge. Opposite charges attract, like charges repel. This simple rule, extended across enormous numbers of particles, governs the structure of molecules. When two atoms approach one another, their electrons begin to interact. The negatively charged electrons of one atom repel the electrons of another, yet each nucleus simultaneously attracts the other atom's electrons. What emerges from this competition is not chaos, but balance. If the arrangement lowers the total energy of the system, the atoms remain bound. If it does not, they separate. Bonding is therefore not a preference, but a consequence of energy minimization under electromagnetic interaction. In DNA, the backbone connecting each nucleotide is held together by covalent bonds. A covalent bond forms when two atoms share electrons. Sharing does not mean an electron sits neatly between them, like a bead on a string. It means that the electron's quantum state extends across both nuclei at once. The attractive pull from both positively charged centers stabilizes this shared distribution. As long as this configuration has lower energy than the separated atoms, the bond persists. The sugar and phosphate groups that form the rails of the DNA ladder are linked through such shared electron states. Carbon atoms bond with oxygen. Phosphorus bonds with oxygen. Each connection arises from the same underlying principle, electromagnetic attraction structured by quantum rules. The shape of the molecule follows from the angles at which these bonds prefer to arrange themselves, and those preferred angles are determined by how electrons distribute in space. Even the rungs of the ladder depend on electric charge. The nitrogen bases that pair across the spiral attract one another through weaker electromagnetic interactions known as hydrogen bonds. These bonds are not as strong as covalent bonds, yet they are strong enough to hold complementary bases together, while still allowing the strands to separate during replication. The difference in strength arises from how electrons are shared, or partially shifted between atoms, creating regions of slight positive and negative charge that draw toward one another. Nothing in this structure requires life to function. The same electromagnetic force that causes static electricity also holds the DNA molecule intact. What makes DNA special is not a new force, but a particular arrangement of charged particles that settles into a stable configuration under these universal laws. The double helix persists because its configuration represents an energetically favorable balance of attractions and repulsions among vast numbers of electrons and nuclei. Stability is the natural outcome when electric forces are arranged in the lowest accessible energy state permitted by quantum mechanics. The bonds that hold DNA together depend on electrons, and electrons do not behave like miniature solid spheres. When early physicists tried to describe them as tiny particles orbiting a nucleus like planets around a star, the model failed. It could not explain why atoms emit light at specific colors. It could not explain why matter remains stable instead of collapsing inward. A deeper description became necessary. Quantum mechanics revealed that electrons possess both particle-like and wave-like properties. They do not travel along fixed paths. Instead, their behavior is described by a mathematical object known as a wave function, which determines the probability of finding the electron in various regions around the nucleus. This probability distribution forms structured patterns in space. These patterns are not arbitrary. They are constrained by strict rules that arise from the quantization of energy. An electron bound to an atom can only occupy certain allowed energy levels. Between those levels, it cannot exist. This restriction prevents the electron from spiraling into the nucleus. It also prevents it from drifting away unless sufficient energy is supplied. The atom therefore has a stable size, because only discrete spatial distributions are permitted. Each allowed distribution corresponds to a specific energy state. These structured distributions are called orbitals. An orbital is not a path, but a three-dimensional region where the electron is most likely to be found. Some orbitals are spherical, others form lobed shapes extending in particular directions. The geometry of these orbitals determines how atoms can connect with one another. When two atoms approach, their outer orbitals interact. If their shapes and energies align in a way that lowers total energy, a bond forms. Inside DNA, every covalent bond depends on the overlap of orbitals between neighboring atoms. The stability of the backbone, the precise angles between atoms, and the planar structure of many of the nitrogen bases all arise from the geometry of these electron distributions. The shape of the molecule is therefore not imposed from outside. It emerges from the quantum mechanical constraints governing electron behavior. What appears at the biological scale as a stable double helix is, at its foundation, the consequence of electrons occupying allowed wave patterns around atomic nuclei. The structure persists because those patterns represent the lowest energy arrangements accessible under quantum law. The quantum behavior of electrons does not remain confined within individual atoms. When atoms approach one another, their outer orbitals begin to overlap. This overlap changes the distribution of electron probability. Instead of belonging exclusively to one nucleus, the electron's wave pattern can extend across multiple nuclei simultaneously. The geometry of this shared distribution determines how atoms arrange themselves in space. Orbitals possess distinct shapes. Some are spherical and extend evenly in all directions. Others form elongated lobes oriented along specific axes. These shapes arise from the mathematical constraints placed on the electron's wave function. Because the wave function must satisfy strict boundary conditions, only certain spatial patterns are permitted. Each permitted pattern carries a specific angular structure. When carbon atoms bond in DNA's backbone, the outer orbitals rearrange into hybridized forms. These hybrid orbitals point in specific directions, allowing bonds to form at characteristic angles. The familiar tetrahedral arrangement around many carbon atoms is not a geometric preference imposed from above. It is the direct result of how electron waves combine to minimize energy, while satisfying quantum constraints. The nitrogen bases inside DNA contain ring structures that lie nearly flat. Their flatness arises because certain orbitals overlap side by side, forming what are known as pi bonds. These bonds create electron density above and below the plane of the atoms, stabilizing the ring and restricting rotation. The rigidity of these bases is therefore a manifestation of electron wave symmetry. The double helix itself reflects the cumulative effect of these microscopic geometries. Bond angles, bond lengths, and planar regions combine in a repeating pattern that naturally twists when extended in 3 dimensions. The twist is not decorative. It emerges from how spatial constraints propagate along a long chain of bonded atoms. What we observe as molecular shape is therefore the visible imprint of invisible quantum structures. The positions of atoms are not arbitrary points in space. They are equilibrium locations determined by how electron wave patterns overlap and settle into their lowest permitted energy configurations. Geometry, at its root, is quantum behavior expressed in 3 dimensions. When 2 atoms in DNA form a covalent bond, something subtle happens at the quantum level. The electron that participates in the bond is no longer confined to a single nucleus. Its wave function spreads across both atoms, creating a shared state. This shared distribution lowers the total energy of the system because the negatively charged electron now experiences attraction from 2 positively charged nuclei at once. The bond persists precisely because this arrangement is energetically favorable. This sharing is not a classical exchange. The electron does not travel back and forth like a courier. Instead, its quantum description encompasses both nuclei simultaneously. The probability of finding it exists in a continuous region that links the atoms together. In this sense, the bond is not a physical thread connecting 2 beads. It is a stable pattern in the underlying electron field that includes both atomic centers within a single coherent structure. In the DNA backbone, carbon atoms bond to oxygen and phosphorus through these shared quantum states. Each bond forms only when the orbitals involved align in energy and orientation. If the energies do not match, no stable overlap occurs. If the overlap does not reduce the system's energy, the atoms separate. Stability therefore arises from strict energetic accounting. Bonds exist because they represent the lowest accessible configuration permitted by quantum mechanics and electromagnetic interaction combined. Within the nitrogen bases, multiple covalent bonds create ring structures whose electrons are partially delocalized across several atoms. Instead of being confined between 2 nuclei, some electrons spread across an entire ring. This delocalization increases stability because the electron density is distributed more evenly, lowering repulsive interactions. The molecule settles into the arrangement that balances attraction and repulsion most effectively. The continuity of the DNA strand depends on millions of these shared quantum states aligned in sequence. Each bond is local, yet together they produce a macroscopic structure capable of storing biological information. The integrity of the molecule does not arise from rigidity alone. It arises from countless electrons occupying stable, shared quantum configurations that collectively resist disruption under normal thermal conditions. What we call a chemical bond in DNA is therefore a specific solution to the quantum equations governing interacting electrons and nuclei. The molecule remains intact because these solutions correspond to energy minima that nature consistently favors. The 2 strands of DNA are not fused together by covalent bonds. If they were, separation during replication would require breaking the primary backbone itself. Instead, the rungs of the ladder are held by weaker interactions known as hydrogen bonds. These bonds arise from subtle imbalances in how electrons are distributed within molecules. In certain covalent bonds, electrons are not shared equally. Oxygen and nitrogen atoms tend to attract electrons more strongly than hydrogen does. When hydrogen is covalently bonded to one of these more electronegative atoms, the shared electron density shifts slightly away from hydrogen. This shift leaves the hydrogen nucleus partially exposed, carrying a slight positive character. Nearby atoms with regions of higher electron density carry slight negative character. The attraction between these partial charges creates a hydrogen bond. Within DNA, adenine pairs with thymine through 2 hydrogen bonds, while cytosine pairs with guanine through 3. The difference in bonding number influences the stability of each pair. 3 hydrogen bonds create a slightly stronger interaction than 2, and this difference affects how tightly certain regions of DNA are held together. Yet even 3 hydrogen bonds are individually weaker than a single covalent bond. This balance allows the strands to remain associated under normal conditions, while still permitting controlled separation when biological processes require it. The geometry of hydrogen bonding depends on precise orientation. The atoms must align so that the partially positive hydrogen sits between electronegative atoms at specific angles. These preferred angles arise from the spatial distribution of electron orbitals. The bond forms only when the arrangement lowers the overall energy of the interacting bases. Once formed, the hydrogen bonds collectively stabilize the double helix without locking it permanently. The pairing rules of DNA, often described in biological terms as complementary matching, are in fact consequences of quantum mechanical charge distributions and molecular geometry. Only certain arrangements allow the correct alignment of partial charges and orbital shapes. All other pairings fail to achieve comparable stability and therefore do not persist. The 2 strands of DNA remain gently bound because electromagnetic interactions between unevenly distributed electrons create directional attractions that settle into a repeating, energetically favored pattern along the length of the molecule. Hydrogen bonds explain how the 2 strands of DNA recognize and hold one another. But the deeper reason the whole structure stays intact is that every part of the molecule sits in a landscape of allowed energies. In the quantum picture, stability is not a vague sense of firmness. It is the fact that electrons and nuclei can only occupy certain states, and that some of those states are lower in energy than others. Once a system settles into a low-energy arrangement, it does not change unless something supplies enough energy to push it out. Inside an atom, electrons are restricted to discrete energy levels. Inside a molecule, the situation becomes richer, but the same principle remains. When atoms bond, their orbitals combine to form molecular orbitals that extend across several nuclei. These molecular orbitals also come in allowed energy states. Some of them are bonding states, where the electron density lies in regions that increase attraction between nuclei and lower the energy. Others are anti-bonding states, where the electron density is arranged in a way that weakens attraction and raises the energy. A stable molecule exists when its electrons predominantly occupy the bonding states that make the total energy as low as possible. DNA's backbone is strong because the electrons in its covalent bonds occupy bonding configurations with a large energy gap separating them from antibonding alternatives. That gap matters. Thermal motion inside a cell constantly jostles the molecule, but random thermal energy usually is not enough to lift electrons into states that would break those bonds. The backbone therefore remains intact under ordinary biological conditions because the energetically accessible states do not include easy pathways to bond rupture. The bases also benefit from this energy structure. Their ring systems have arrangements of electrons that are especially stable, partly because electrons can spread across the ring, rather than being confined to a single link. This delocalization lowers energy and creates rigidity. The bases keep their shapes because twisting or distorting them would force electrons into less favorable configurations. Molecular stability is therefore partly a matter of geometry, but geometry itself is enforced by which electron states are energetically permitted. Even the overall double helix reflects this principle. The helix is not held by one single lock. It is held by the fact that many small interactions together create a minimum energy arrangement for the whole molecule in its environment. If the strands separate, hydrogen bonds are disrupted and the energy rises. If the strands pair correctly and stack in the helix, the energy falls. DNA remains in its familiar form because it naturally occupies the configuration that costs the least energy while satisfying quantum constraints. So when we ask what DNA is made of according to quantum physics, one part of the answer is that it is made of matter that has found an energetically favored set of quantum states. Its persistence is the quiet consequence of electrons occupying stable, quantized configurations that keep the molecule in a low energy shape. Even in its most stable configuration, DNA is not motionless. Inside a living cell, temperature ensures constant molecular movement. Atoms vibrate, bonds stretch and compress, segments of the double helix flex slightly. This motion does not contradict stability. It exists within it. Thermal energy is the collective result of countless microscopic interactions. At body temperature, molecules possess enough energy to move and vibrate, but not enough to disrupt the strongest covalent bonds in DNA's backbone. The quantum energy gaps that separate bonding from antibonding states act as boundaries. Thermal fluctuations can excite small vibrations within a bond, but they rarely supply the large, specific energy required to break it. Stability therefore coexists with motion because the allowed energy landscape restricts what fluctuations can accomplish. These vibrations are themselves quantized. The bonds in DNA can stretch or bend only in certain vibrational modes. Each mode corresponds to a discrete energy value, much like electrons in atoms occupy discrete levels. The molecule can absorb or release energy by moving between these vibrational states, but it cannot vary continuously in any arbitrary way. The motion of DNA is therefore structured, not random in the classical sense. It unfolds within defined quantum patterns. The hydrogen bonds between base pairs are weaker, and thermal motion sometimes causes brief local separations. A base pair may momentarily open and then close again, as surrounding interactions restore the lower energy configuration. This flexibility is essential for biological processes such as replication and transcription. Yet even here, the motion remains constrained. Only certain distortions are energetically accessible under normal conditions, and the overall structure persists because returning to the paired state lowers energy once more. The entire molecule behaves like a dynamic system oscillating around a minimum energy arrangement. It is not frozen. It is not fragile. It is a balance between movement and restriction. Quantum rules define which vibrational patterns are possible, and electromagnetic forces determine how strongly each deviation is resisted. DNA endures because its structure occupies a basin in the energy landscape, deep enough to withstand ordinary thermal agitation. Motion is present everywhere within it, but that motion unfolds inside boundaries set by quantized energy levels and fundamental forces. Within the nitrogen bases of DNA, something particularly important occurs at the quantum level. The electrons involved in certain bonds are not confined between just two atoms. Instead, they are shared across an entire ring structure. This spreading of electron density is known as delocalization. It is not a vague cloudiness. It is a precise quantum state in which multiple atomic orbitals combine into a larger continuous distribution. The electrons in the double bonds of these rings occupy molecular orbitals that extend above and below the plane of the atoms. Rather than belonging to a single bond, these electrons are distributed across the ring as a whole. This arrangement lowers the total energy because the electron density is not forced into a narrow region where repulsion would increase. Spreading out stabilizes the structure. That stability has consequences beyond mere rigidity. The specific shapes of the bases determine how they pair. Their edges present patterns of partial positive and negative charges that arise directly from how electrons are distributed within the ring. If the delocalized electrons were arranged differently, the charge pattern would change, and the pairing rules would not hold in the same way. The reliability of base pairing is therefore anchored in the quantum mechanical structure of these electron states. Delocalization also contributes to stacking interactions between neighboring bases along the helix. The flat rings tend to align above one another. When they do, their extended electron distributions interact through electromagnetic attraction and subtle repulsion, creating an additional stabilizing effect. The double helix is not held together only across the rungs, but also along its length by these stacking interactions. The repeating order of bases therefore enhances structural coherence. Because the electron distributions in the bases are energetically favorable and geometrically constrained, the molecular shapes remain consistent over time. The information encoded in the sequence of bases persists because the underlying quantum states that define each base are stable under ordinary conditions. A change in information requires a change in molecular structure, and such a change requires sufficient energy to rearrange these electron configurations. The apparent permanence of genetic information thus rests on the stability of delocalized quantum states within each nucleotide. DNA stores information reliably because its electron structures occupy energetically preferred arrangements that resist spontaneous rearrangement. Within each hydrogen bond that connects the bases of DNA, a single proton plays a delicate role. That proton sits between two electronegative atoms, attracted to both. In classical physics, we might imagine it as a tiny particle resting in a small valley of energy, positioned closer to one atom than the other. Quantum mechanics refines this image. The proton does not possess a perfectly fixed location. Its position is described by a probability distribution shaped by the energy landscape of the bond. In most situations, this distribution is strongly weighted toward one side. The proton is far more likely to be found near the atom to which it is covalently bonded. That asymmetry preserves the usual base pairing structure. Adenine pairs with thymine, cytosine pairs with guanine. The hydrogen bonds align in specific orientations, maintaining the familiar geometry of the double helix. Yet quantum mechanics allows a subtle possibility. If the energy barrier separating two nearby configurations is not infinitely high, there exists a small probability that the proton can appear on the other side without climbing over the barrier in the classical sense. This process is known as quantum tunneling. It does not violate conservation laws. It arises because the proton's wave function extends slightly into regions that would be forbidden if it were treated purely as a classical particle. In DNA, proton tunneling within hydrogen bonds can occasionally shift the bonding pattern of a base pair. When that happens, the geometry changes slightly, and the pairing preferences may alter temporarily. Most of the time, such shifts are brief and reversible. The system relaxes back into its lower energy configuration. The dominant quantum state remains the one that preserves standard base pairing. On rare occasions, however, If replication occurs while a proton occupies this shifted configuration, a mismatch can be introduced. This mechanism is one of several subtle pathways through which mutations may arise. Even here, the process is not mysterious. It is governed by the same quantum principles that define electron distributions and energy levels. The probability is small because the energetically favored configuration strongly dominates, but it is not zero. DNA's reliability therefore coexists with quantum possibility. The molecule is overwhelmingly stable because its lowest energy states are strongly preferred, yet the probabilistic nature of quantum particles allows rare transitions that introduce variation. The structure persists because the dominant configurations are energetically secure, while the underlying quantum framework permits minute fluctuations within those boundaries. The presence of quantum tunneling might suggest that DNA behaves like a delicate quantum system in the same sense as particles isolated in laboratory experiments. Yet DNA exists in a warm, wet cellular environment. It is constantly interacting with surrounding water molecules, dissolved ions, and nearby proteins. These interactions matter because quantum systems maintain delicate phase relationships only when they are shielded from disturbance. In ordinary biological conditions, such isolation does not exist. Quantum coherence refers to the preservation of precise phase relationships within a wave function. In carefully controlled systems, coherence allows interference effects that reveal the wave nature of matter. In DNA, electrons and protons still obey quantum mechanics, but large-scale coherent superpositions do not persist for long. The environment interacts continuously with the molecule, effectively measuring and disturbing delicate phase relationships. This process, known as decoherence, suppresses large-scale quantum interference. That suppression does not remove quantum behavior. It constrains how it manifests. Electrons still occupy quantized orbitals. Energy levels remain discrete. Tunneling probabilities still arise from wave function behavior. What disappears rapidly is long-lived coherence across large regions of the molecule. Instead of acting as one giant quantum object, DNA behaves as a collection of smaller quantum systems whose states are continually stabilized by environmental interaction. This stabilization has a practical effect. It ensures that the molecule behaves reliably at biological scales. If large coherent superpositions were easily sustained, molecular configurations might fluctuate in unpredictable ways. Decoherence instead forces the system into well-defined, localized states that correspond to stable chemical structures. The environment effectively reinforces classical-like stability, while the underlying quantum rules continue to govern allowed states and transitions. DNA therefore exists in a balanced regime. At the smallest scale, electrons and protons follow quantum laws exactly. At the scale of the entire molecule, interactions with the environment suppress fragile superpositions and preserve consistent structure. The reliability of genetic information depends not on exotic coherence, but on quantum mechanics operating within conditions that favor stable, localized configurations. Up to this point, DNA has resolved into atoms bound by electromagnetic forces, with electrons occupying quantized states that determine structure and stability. Yet even atoms are not indivisible. The nucleus at the center of each atom contains protons and neutrons, and those particles themselves possess internal structure. Protons and neutrons are not fundamental in the deepest sense. They are composed of smaller constituents known as quarks. Quarks carry fractional electric charge and interact through a force far stronger than electromagnetism at very short distances. This interaction is mediated by particles called gluons, which bind quarks together inside protons and neutrons. The strength of this binding is extraordinary. It is the reason the nucleus remains intact, despite the electrical repulsion between positively charged protons. The mass of DNA arises largely from these protons and neutrons. Electrons contribute only a small fraction of the total mass. When you consider the weight of a strand of DNA, nearly all of it comes from the energy bound up inside atomic nuclei. That mass is not simply stored substance. According to relativity, mass corresponds to energy. The binding energy that holds quarks together contributes significantly to the mass of the proton itself. Inside every carbon, oxygen, nitrogen, and phosphorus atom in DNA, quarks are confined within protons and neutrons by the strong interaction. That confinement is stable because the force grows stronger as quarks attempt to separate. They remain locked together, forming the nucleons that in turn form atomic nuclei. The nucleus provides the concentrated positive charge that organizes electron distributions around it. The electromagnetic force shapes chemistry. The strong force stabilizes nuclei. Both operate simultaneously within every atom of DNA. The molecule that carries genetic information is therefore built from structures governed by at least two fundamental interactions working in concert. Without the strong force, nuclei would not exist. Without electromagnetism, electrons would not bind to nuclei. Without quantization, no stable energy levels would form. DNA, when traced inward to this depth, is composed of electrons, quarks, and the forces that govern them. Its familiar biological role rests upon layers of interaction that begin far below the scale of molecules. What appears as a stable, repeating double helix is ultimately supported by confined quarks inside protons and neutrons, and by electrons arranged in quantized states around them. Quarks and electrons may seem like the final pieces, yet modern physics describes even these particles as manifestations of something deeper. According to quantum field theory, what we call a particle is not a tiny solid object existing independently in space. It is a localized excitation of a field that extends everywhere. An electron is an excitation of the electron field. A quark is an excitation of its corresponding quark field. The electromagnetic force arises from interactions in the electromagnetic field. These fields are not inserted into space. They are the underlying structure of space itself. At every point in the universe, these fields have values. Under certain conditions, those values oscillate in stable, quantized ways. We observe those oscillations as particles. When you imagine an electron in a DNA molecule, what truly exists is a stable excitation of the electron field interacting with excitations of quark fields confined within protons and neutrons. The nucleus is a region where quark and gluon fields are intensely bound. The surrounding electron distributions are structured patterns in the electron field shaped by electromagnetic interaction. The molecule as a whole is a highly organized arrangement of interacting fields settling into a stable configuration. This perspective does not remove the reality of atoms or molecules. It explains them more deeply. Atoms are persistent patterns in interacting quantum fields. Molecules are larger patterns formed when those atomic configurations minimize their combined energy. DNA is one such pattern, extended in a repeating sequence that remains stable because the underlying field interactions support that arrangement. The solidity you would feel if you could touch an enlarged strand of DNA arises from electromagnetic field interactions resisting compression. The mass of the strand arises largely from energy bound within quark and gluon fields inside nuclei. The bonds arise from structured overlaps of electron field excitations. Nothing in this description requires substance in the classical sense. What exists are fields and stable vibrations within them. DNA, traced to its deepest level known in modern physics, is an organized, low-energy configuration of quantum fields interacting according to precise laws. The molecule persists because this configuration satisfies those laws while minimizing total energy under the conditions present inside a cell. When we speak of DNA, we often speak of information. We describe sequences as instructions, as codes, as messages passed forward through generations. Yet nothing in the quantum fields themselves contains intention or language. What exists are stable configurations of matter and energy. Information emerges because certain configurations can be distinguished, copied, and preserved. Each nucleotide base differs slightly in structure. Adenine is not identical to thymine. Cytosine is not identical to guanine. These differences arise from distinct arrangements of atoms, which arise from distinct solutions to quantum mechanical constraints. Because the electron distributions and nuclear compositions differ, the shapes and charge patterns differ. Those physical differences make each base chemically recognizable. When DNA replicates, enzymes interact with these bases through electromagnetic forces. They do not read symbols. They respond to geometry and charge. Complementary pairing occurs because the quantum mechanically stabilized structures of the bases allow certain hydrogen bonding patterns and exclude others. The sequence along one strand determines which bases can attach to it during replication. In this way, physical compatibility gives rise to reliable copying. Information, then, is not an added layer placed on top of matter. It is a pattern in matter that can be distinguished from alternative patterns and reproduced through lawful interaction. The reliability of that pattern depends on the stability of the underlying quantum states. If the electron configurations of the bases fluctuated wildly, consistent pairing would not occur. If nuclear structures were unstable, atoms would not persist long enough to form molecules. The endurance of information is anchored in the endurance of the physical arrangements that embody it. The sequence of bases along DNA is therefore a sequence of stable quantum mechanical structures arranged in a specific order. That order persists because each unit resists spontaneous rearrangement under normal conditions. Replication is possible because complementary structures lower energy when brought together. Variation arises rarely because quantum probabilities permit occasional deviations within tightly constrained limits. What appears as a genetic code is, at the deepest level, a sustained pattern of quantum field excitations arranged in a sequence that can be physically copied. Information emerges because stable differences in structure can be maintained and reproduced within the laws of physics. If we now gather everything together without stepping backward, a continuous structure becomes visible. DNA is not made of life as a separate substance. It is made of nucleotides arranged in a repeating chain. Each nucleotide is a molecule built from atoms. Each atom contains a nucleus surrounded by electrons in quantized states. Each nucleus contains protons and neutrons composed of quarks, bound by the strong interaction. Each electron and quark is an excitation of an underlying quantum field. At every layer, stability arises from the same principle. Systems settle into the lowest energy configurations permitted by fundamental laws. Electron orbitals form because only certain wave patterns are allowed. Covalent bonds persist because shared quantum states lower total energy. Hydrogen bonds stabilize base pairing because aligned charge distributions reduce energy further. Vibrational motion remains limited because only discrete energy transitions are permitted. Nuclear structure endures because the strong interaction confines quarks into stable combinations. The double helix is therefore not an imposed design. It is the macroscopic expression of countless microscopic constraints aligning simultaneously. Bond angles, charge distributions, orbital overlaps, energy gaps, and field interactions converge into a repeating geometry that persists under ordinary biological conditions. The shape of DNA is the natural outcome of these layered constraints operating together. The information stored along its length does not float above physics. It is encoded in the physical differences between stable quantum mechanical arrangements. The reliability of inheritance depends on the fact that these arrangements are energetically favored and resistant to random disruption. Variation occurs because quantum probabilities allow rare transitions, yet the dominant configurations remain overwhelmingly preferred. From the scale of fields to the scale of cells, nothing in DNA escapes the laws that govern all matter. What makes DNA extraordinary is not a special exemption from physics, but the way ordinary physical principles assemble into a structure capable of persistence and replication. The molecule exists because quantum fields interacting through fundamental forces permit this particular organized pattern to occupy a stable, low-energy configuration. When we ask what DNA is made of according to quantum physics, the answer is coherent across every scale. It is made of stable excitations of quantum fields arranged into atoms, assembled into molecules, organized into a double helix, whose structure follows inevitably from the allowed interactions of charged particles. That layered stability is what ultimately holds the molecule, and the information within it, in place. A structure can be stable in an instant and still fail over time. For DNA to serve its biological role, its quantum mechanical foundation must remain intact amid constant interaction. Inside a cell, collisions occur continuously. Water molecules strike its surface. Ions drift nearby. Proteins bind and release. Each encounter perturbs the electromagnetic environment, yet the molecule rarely loses its structure because the energy barriers protecting its covalent backbone are substantial. Random collisions distribute energy briefly and unevenly, which is usually insufficient to break those bonds or rearrange the stabilized electron configurations that define them. DNA does not sit motionless within this activity. It bends, twists, and its base pairs momentarily open and close. These motions occur within a structured energy landscape shaped by quantized states. The molecule fluctuates inside a basin of stability. When small distortions occur, electromagnetic forces naturally guide the system back toward lower energy configurations. Stability arises not from rigidity, but from the persistent preference for energetically favored arrangements, permitted by quantum rules. Damage also follows lawful pathways. Ultraviolet radiation can excite electrons into higher energy states, and if sufficient energy is absorbed, bonds may rearrange. Such transitions occur only when discrete energy thresholds are crossed. The structure does not degrade arbitrarily. It changes only under specific quantized conditions defined by the same principles that created it. Across longer timescales, replication sustains continuity. Complementary bases pair because their quantum-stabilized geometries and charge distributions align in consistent ways. Each successful copy reinstates the same low-energy molecular pattern. Persistence through generations is therefore an extension of energy minimization operating within a constrained quantum framework. Tracing inward, atoms resolve into nuclei and electrons, and these into quarks and fields. Electrons are excitations of the electron field. Quarks are confined by the strong interaction inside protons and neutrons. The electromagnetic field governs attraction and repulsion, organizing atoms into molecules. DNA exists because these interacting fields can assemble into a stable, repeating configuration under cellular conditions. At the deepest level currently accessible to experiment, DNA is not made of a special biological substance. It is a structured pattern of quantum field excitations arranged into atoms, assembled into molecules, organized into a helix capable of replication. What we call genetic information is a sequence of physically distinct, quantum mechanically stabilized configurations arranged in order. DNA is organized energy held in place by fundamental interactions. The same laws that govern stars, electrons, and nuclei also sustain the code of life with quiet consistency. And so, when we ask what DNA is made of according to quantum physics, the answer is both simple and profound. It is made of the same fields and forces that shape the rest of the universe. In one quiet corner of those laws, energy has arranged itself into a pattern stable enough to remember, to copy, and to endure. The code of life is not separate from physics. It is physics, settled into form.